My study focused on ankle-foot orthoses and their impact on ankle kinematics in adults with cerebral palsy. Cerebral palsy is a childhood-onset neurodevelopmental condition that affects muscle coordination, movement, and posture. Because of this, the ankle joints often experience improper loading. For this project, we focused on the tibiotalar and subtalar ankle joints. Previous studies have found that cerebral palsy may be correlated with a higher risk for early-onset osteoarthritis. While the exact reason for this isn't definitively known, it's clinically hypothesized that irregular gait, uneven weight distribution, and joint misalignment may lead to accelerated cartilage breakdown in the joints. A clinical intervention that aims to correct these improper foot biomechanics and prevent the progression of osteoarthritis is the ankle-foot orthosis. These are external braces worn inside the shoe to correct foot positioning, improve ankle stability, and improve gait. They are non-invasive and come in a variety of different types and customizations depending on the individual. Our study compared two types: hinged AFOs, which allow dorsiflexion but restrict plantar flexion to 90 degrees, and semi-rigid AFOs, which permit some dorsiflexion and plantar flexion but offer a lot more stability. However, there is currently limited evidence showing that AFOs actually improve biomechanics in adults with cerebral palsy. So why is there this gap in knowledge? For one, cerebral palsy is a childhood onset condition, so a lot of the research out there focuses on preventative measures during childhood. Additionally, up to 58% of AFO prescriptions may be inadequate, so AFO disuse is pretty common in adults. To address this, our study focused on an adult population of individuals with cerebral palsy. Previous studies have also used skin marker motion capture, but this method of data collection is a little bit limited because it cannot distinguish between the two ankle joints and instead views them as one joint complex. To address this, we use dual fluoroscopy, which consists of two orthogonal X-ray setups. In each setup, an X-ray source emits X-rays, which are then captured by an intensifier and camera. The system lets us directly view in vivo bone motion in the combined field of view and independently assess the tibiotalar and subtalar ankle joints with under a millimeter and under a degree of error. Our objective was to compare tibiotalar and subtalar ranges of motion across three conditions, AFO, shod, and barefoot, in adults with cerebral palsy using dual fluoroscopy. We hypothesized that the AFO would reduce dorsiplantar flexion in the tibiotalar joint and minimally affect the subtalar joint. We recruited 4 adults with cerebral palsy and imaged them walking barefoot, shod, and with their individually prescribed AFOs. We also recruited 5 controls and imaged them walking barefoot. We collected dual fluoroscopy images during stance phase at a capture rate of 120 Hz. We then obtained CT scans to create 3D bone models of the tibia, talus, and calcaneus. Each of the bone models were then aligned to the images to triangulate their position in 3D space. Doing this for each bone and frame allowed us to visualize how the bones were moving spatiotemporally. From this data, tibiotalar and subtalar joint angles were calculated and averaged across participants. Ranges of motion were found as the difference between maximum and minimum joint angles. Statistical analysis showed no significant differences in range of motion between conditions in either joint. However, we observed a general trend. For the tibiotalar joint, the AFO condition had the lowest mean range of motion, which aligned with our hypothesis, although it was not statistically significant. The subtalar joint showed a similar trend where the AFO had the lowest mean range of motion, although again this was not statistically significant, and this reduction in range of motion was a lot more minimal. When we looked at the waveforms over time, the AFO condition appeared to limit plantar flexion almost completely in both joints, which is consistent with the prescribed purpose of the AFOs used in this study. However, averaging data across participants hid a lot of individual variation. Comparing the mean AFO waveform in bold to each individual participant showed just how much information was lost by group level averaging. This brings me to the biggest key takeaway from this study, which was that group level averaging may not be appropriate for this population. In the future, we could look at individual case studies that take into account foot deformity, AFO type, and AFO prescription. Our small sample size was another limitation, so having a larger sample size in the future would allow us to make more confident generalizations based on results. Although our results weren't statistically significant, they suggest that AFOs may primarily restrict plantar flexion in the sagittal plane during stance phase. In the future, we could look at other planes of motion, as well as image swing phase. However, the non-significance we observed could also be an indication that the functional benefits of AFOs are independent of range of motion and joint angles, for example, improving balance and reducing energy expenditure. Overall, future studies using dual fluoroscopy to track in vivo bone motion with an AFO have the potential to provide clinical insights into AFO prescription. Here are my references. And thank you so much to my lab and everyone who helped out with this project, as well as Alberta Innovates for funding my research this summer.